2 12th and one each from 10th and 11th. And topic will be given 3 minutes before your speech. Okay. So good afternoon, students.
Good afternoon, sir.
Yeah,
Good
yeah,
afternoon, sir.
Good
yeah,
afternoon, sir.
good afternoon.
Good
We'll
afternoon,
continue.
sir.
Yeah. So we'll continue with the topics that we are discussing in the morning. We were discussing about the structure of proteins. Okay. And the structure of proteins were discussed in 4 different levels: primary, secondary, tertiary, and quaternary. We'll have a quick glance through that. Um Primary structure, it's a sequence of amino acids. Secondary structure, shape in which a long polypeptide chain is existing, two forms, alpha. Uh, it exists where R group is large, right-handed screw structure. NH and CO form a hydrogen bond, also known as 3.6 13 helix, since in a turn of a helix, approximately 3.6 amino acids and 13 membrane is formed by hydrogen bonding. CONH group is peptide bonds, and all these things. Beta peptide exists when R group is small. Peptide chains are stretched out to maximum, and then they lay side by side and held together by hydrogen bonds. Tertiary structure represents the overall folding of polypeptide chain, and it is stabilized by disulfide linkages, hydrogen bonding, ionic bonds, okay, or we can Coulombic interactions, or and van der Waals forces. Two shapes are there: fibrous and globular, as a result of tertiary structure. And quaternary structure is the overall, the number of subunits and overall configuration or arrangement of the subunits with respect to each other. That is the that is meant by the quaternary structure. Okay. So that is what we have seen, and you can see that whenever a protein is having a unique three-dimensional structure, and when it performs its biological activity, whenever it is having the three-dimensional structure, unique structure, and when it performs the activity, we call it as a native protein. Okay, we call it as a native protein. A protein having a three-dimensional structure, and it will be unique for that protein, and performs its biological activity. Okay, activity, that is what we call as a native protein. And you can see that any changes in the physical state of the protein makes it inactive. Okay, if the secondary and tertiary structure are destroyed, if the secondary or if the hydrogen bonds are broken, if the coiling is unfolded, then they will lose their secondary and tertiary structure, and that will lead to the denaturation of protein. Denaturation of protein. And as a result of denaturation of protein, they will lose their biological activity. Okay, so whenever there are certain uh, temperature and the pH at which their activity is maximum, and you can see that whenever we change the pH, or when we increase the temperature, the hydrogen bonds will be broken, and unfolding of the coils will take place, and as a result of unfolding folding of the coils, they will be losing their tertiary and secondary structure. And when they lose their tertiary and secondary structure, they will lose their uh, biological activity. Biological activity. Okay, that process of making a native protein inactive by increasing the temperature or by changing the pS, by which tertiary and secondary structures uh, are broken, or tertiary and secondary structures are destroyed. That process is what we call as the denaturation of protein. The best example of denaturation is boiling of egg. Okay, so when egg is boiled, what happens? It becomes a hard material. It becomes that a white part. You get a hard white part, and that is nothing but the denaturation of protein. That fleshy part is proteinous, but once it is boiled, it becomes hard. That is example. Or curding of milk. Curding of milk is another example of denaturation of protein. Okay, and denaturation is nothing but the process of making a protein inactive, the loss of biological activity of a protein. When protein is subjected to physical change like temperature or chemical change is known as denaturation. Okay, so that is all about the proteins. Now let me ask you, any doubts regarding proteins? Any doubts regarding proteins? During denaturation, there is no change in the primary structure. Primary structure remains intact. That means the sequence in which amino acids are arranged, that remains intact, but secondary and tertiary structures will be destroyed. Okay. Any doubts? So you should be thorough with the structure of protein and what is a polypeptide and what is the linkages between two amino acids. Okay. And why amino acids are amphoteric? Why amino acids are amphoteric?
It has NH2 group and carboxylic group, which is acid and base.
Yeah, or we will say it is not as such due to the presence of NH2 and COH. It is due to the formation of Sitter ion. Sitter ion. What is Sitter ion?
Which is both positive and negative
Yeah,
ions, which
the
has both
internal
positive.
salt.
Both positive and negative.
Yeah, we call it as a
Dipolar.
dipolar ion or an internal salt formed due to the reaction between the acidic group and the amino group. So it is possible in case of amino acids, and it will have both the cationic and anionic part. That is why it will react with both acids as well as bases and behaves as a amphoteric material. Amphoteric material. Okay, so that is all about the proteins, and we had enzymes which is removed from deleted, and we have the enzyme mechanism. Anyway, you have already studied about these enzymes in the surface chemistry, but it is not there. uh, that enzyme action, all these are not there in the biomolecules. It is removed. Then vitamins were there, and all these vitamins are removed, so that also will not discuss. You want me that discuss that? Not required actually. Okay, then hormones were there, that hormones also removed, and the remaining one topic is nucleic acids. We are going to discuss about another very important topic, which is nucleic acids. Okay, let me ask you, you will be very much familiar with the nucleic acids. Praveen, 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 or Srikanth? So if I am asking, nobody will open your mouth, is not? Vishwaharan? Vishwaharan?
Yes, sir.
So what is the role of nucleic acid in living systems? Can you tell me?
Sir, it is present in DNA.
Present in
DNA.
nucleic acid is present in DNA, only in DNA?
RNA, sir.
RNA. DNA and RNA. Can you hear me?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Network is
Yes, sir.
lost. It's okay.
Yeah. You will get disconnected after some time now, sir. It is showing like this.
Okay. Now you are audible? Now I'm
Yes,
audible
sir.
for you?
Yes,
Yeah.
sir.
Yeah. Good. Now the material which is responsible for
Yes,
Yes,
sir.
sir.
Yes, sir.
okay, good. So we were discussing about these chromosomes. So the material which is present in the nucleus of a cell, which is responsible for transmission of heredity, is chromosomes. And these chromosomes are generally made of two things. One is a nucleic acid, the other one is a protein. Okay. So nucleic acid and proteins, and these nucleic acids are uh, either DNA or RNA. Nucleic acids are either DNA or RNA. DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. Deoxyribonucleic acid. And other one will be ribonucleic acid, which is RNA. So this is DNA and this one is RNA. So these are the two important types of uh, nucleic acids. Okay. And when you're talking about a nucleic acid, either it is a DNA or RNA, when you're taking the chemical composition of DNA and RNA, it will consist of three parts. Nucleic acid will have three components. Okay. And these three components will be, you will have a pentose sugar, then you will have a nitrogen
Phosphate.
base, then third one, there will be a phosphoric acid group. So these are the three components of a nucleic acid. There will be a pentose sugar, there will be a nitrogen base, and also you will have a phosphoric acid. Okay. Now, so these are the three things. You'll have a phosphoric acid group, then you'll have a sugar, then you'll have a nitrogen base. Okay. And when you are talking about the sugar present in DNA or RNA, DNA will have deoxyribose sugar, whereas RNA will have ribose sugar. And these sugars are nothing but pentoses, 5-carbon sugars. Okay, and we'll draw the structure of these both ribose and deoxyribose. So if I am talking about ribose, it will be having a 5-membered ring. In that one of the position will be occupied by oxygen. The other 4 positions will be carbon atoms. And you have this structure for ribose. CH2OH. So this is first carbon, second carbon, third carbon, fourth carbon, as well as fifth carbon. These are the 5 carbon. So pentose sugar. Will it be alpha form or beta form, this pentose? OH group is on the first carbon is at the top. So that means is it alpha or beta form?
Beta.
This is beta. And this is beta D ribose. It will have D configuration and is beta D ribose. And deoxyribose. And actually the name is beta D2 deoxyribose. Deoxyribose. That means in the ribose at the 2 position, deoxy, that means no oxygen is present. No oxygen is present. So I am drawing the structure again. Here OH, H. Second position, this oxygen will be removed. So instead of oxygen, you will have 2 hydrogen. And the rest of the groups remain same in the other carbon atoms. CH2OH. And this is deoxyribose. No oxygen, without oxygen. This is ribose. And this ribose sugar is the pentose sugar present in RNA, and deoxyribose is present in DNA. Are you getting the difference? The pentose sugar will be deoxyribose, in which case DNA, and it is ribose in RNA. Are you getting? Are you getting these things?
Yes, sir.
Yeah.
Yes, sir.
So yeah. So first difference between RNA and DNA is in the pentose sugar. Is in the pentose sugar, and in case of DNA, it is deoxyribose sugar, whereas RNA it is ribose sugar. Anybody else asking anything?
Sir, what you wrote that BO before the ribose?
Beta D glucose. Where there when we are discussing about the configuration of the compounds?
No,
No.
sir.
So for those boys who were attending this thing, what is that? SSB class. This is the thing. Whenever we are taking a compound, whenever we are talking about asymmetric carbon, okay, asymmetric carbon, we compare that compound with the glyceraldehyde. This is glyceraldehyde. And if the OH group is coming on the right hand side, it will be assigned the configuration D. D plus glyceraldehyde, which means that the OH group is on the right and it is showing dextrorotatory specific rotation. And one more configuration is there, CHO, C, OH, H, CH2OH, where it is known as it is L minus glyceraldehyde. L minus glyceraldehyde. Now, if you look at this one, when you compare the last asymmetric carbon of your compound with that of glyceraldehyde, if it is on the right hand side, it is given the configuration D, and if it is on the left hand side, it is given the configuration L. So, for example, here you see so I am taking the structure of glucose. This is a structure of glucose. This is a structure of glucose. Okay. Now here, this is not the structure of glucose. This is the structure of glucose. Now here, if you are talking about the open chain structure of glucose, here, this open chain structure, first carbon, second carbon, third carbon, fourth carbon, fifth carbon, 6 carbons are there. Fifth carbon is an asymmetric carbon. And where will you have the presence of OH group? Where will you have the presence of OH group? It is on the right hand side. It is comparable with the D-glyceraldehyde. Therefore, it is given the configuration D. And if you OH group on the last asymmetric carbon, even if it is on the left hand side, instead of this OH group on the right hand side, if it is on the left hand side, it is comparable with the L-glyceraldehyde. So it is given the configuration L. Okay. Now another one is alpha and beta. Now you take this one. The first asymmetric carbon. Okay. Now this is alpha and beta. OH group is on the right hand side on the first carbon, which is the anomeric carbon. First asymmetric carbon is known as anomeric carbon, and if OH group is on the right, it is given the configuration. It is called as alpha form of glucose, and if OH group is on the left hand side, it is beta form of glucose. And when you are writing these special arrangements in a compound, whatever is coming on the right hand side, it will come at the top of the line. Here, when you are representing in the pyranose form, this is what we call as a pyranose form of glucose. These two, which is analogous to pyran, which is having a 6-member ring containing one oxygen as a member of the ring. So this is pyranose form. Whatever is coming on the right hand side, it is coming at the bottom of the line, bottom of the bond, and which is whatever coming on the left hand side, that is will be at the top of the line. That is how we are representing these two molecules. Okay. Now, if in the first carbon is asymmetric carbon, there if OH is on the left, that means it which comes at the top, it is beta form. If OH is at the bottom, it is on the right, so it is alpha form. So we'll come back to our molecule. We'll come back to our molecule. This is ribose. This is actually a ribose molecule. In the ribose molecule, in the first carbon, OH group is on the top, that means it is on the left, that means it is beta, and the second carbon, OH group is present, so it is ribose. If oxygen is removed, it will become deoxyribose. It will become deoxyribose. Are you getting? Are you getting?
Yes, sir.
Yeah. Now that is the first difference between RNA and DNA. The base sugar present will be ribose in case of RNA and deoxyribose in DNA. But the structure is not important for you. Just know, should know the difference. The second one is that they have a nitrogen base. Okay, they will have a nitrogen base, and these nitrogen bases are derivatives of purines or pyrimidines. So purines or pyrimidines are heterocyclic nitrogen containing compounds. Heterocyclic compounds containing nitrogen. So this is the purine. Here I have given the structure of pyrimidine. Okay. Then you have adenine and guanine, which is a derivative of purine. You see the difference. Here NH2 is removed. This NH2, here adding an NH2 group at the 6th position of purine, so it becomes adenine. And if at the 6th position you have a double bond O, and if you have an NH2 group at the 2nd position, then it becomes adenine. So you will have two purine nitrogen bases, which is called as adenine and guanine. And these are pyrimidine bases, which is three different derivatives of pyrimidine, where at the 4 carbon you are having an NH2 group, and at the 2nd carbon you have a carbonyl group, then it is cytosine. Then at the 4th carbon you have a carbonyl group, and the 2nd carbon you have a carbonyl group, it becomes uracil. Then at the 4th carbon you have a carbonyl group, 2nd carbon another carbonyl group, and at the 5th carbon if you have a methyl group, it becomes thiamine. So these are the 5: adenine, guanine, cytosine, uracil, and thiamine. These are the 5 nitrogen bases, or we call it simply called as bases. Since it is a heterocyclic compound containing nitrogen, we call it as a base. So these are the 5 bases present in the nucleic acids. And out of these 5, only 4 will be present in a given type. That means in RNA as well as DNA, only 4 will be present. And if you are talking about a DNA, you will have 4: adenine, guanine, thiamine, cytosine, and thiamine. Are you getting? And in case of RNA, the first 3 will remain same, and instead of thiamine, you will have uracil. So that is the difference, the second difference between DNA and RNA. The nitrogen bases in DNA will be adenine, guanine, cytosine, and thiamine, whereas in RNA it will be adenine, guanine, cytosine, and uracil. Are you getting the difference? Are you getting the difference?
Yes.
So
Yes, sir.
you will have, you should make it 2 things clear. One, they will have 3 components. Where sugar component will be there in nucleic acid, every nucleic acid, whether it is DNA and RNA, and there will be 4 nitrogen bases. Actually, 5 nitrogen bases are there. Out of 4 will be present in DNA, and 4 will be present in RNA. In case of DNA, the nitrogen bases will be adenine, guanine. Just learn the name, no need of learning the structure. Adenine, guanine, cytosine, and thiamine will be present in DNA, and adenine, guanine, cytosine, and uracil will be present in RNA. That means instead of thiamine, you will have uracil in RNA. So that is the second difference. Okay, that is the second difference between DNA and RNA. Now, this nucleic
Sir, in RNA, thiamine is not there.
RNA thiamine is not there. Instead of that, instead of thiamine, you will have uracil. Only 4 nitrogen bases will be present, not 5. Okay. You are having a total of 5. Out of that, 4 only will be present in a given nucleic acid. If it is DNA, it will be 1, 2, A, G, C, T. Adenine, guanine, cytosine, and thymine. And if it is RNA, it will be A, G, C, U. Understood?
Yes, sir.
Yeah. So that is the second difference between this one. And normally, when you are talking about this ribose sugar, you can see that it is numbered as 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime, because these nitrogen bases will be numbered as 1, 2, 3. So here the number of atoms are represented 1, 2, 3, 4. Okay, so that is why the ribose sugar will be represented as 1 dash or 1 prime, 2 prime, 3 prime, and 4 prime like this. Now, a base will be attached to the sugar at the 1 prime position. That means at this carbon, instead of this OH group, you will have the base. Instead of OH group, you will have the presence of base. And that combination of a pentose sugar and a base at the 1 dash position of the carbon or the sugar moiety, that is what we call as a nucleotide. That is what we call as a nucleotide. So what is a nucleotide? It is a pentose sugar plus nitrogen base. Where is this nitrogen base attached?
At
Nitrogen
1
base
prime.
is at the 1 prime position of the sugar. Nitrogen base is attached at the 1 prime position of the sugar. Then at the 5th position, if a phosphoric acid group is attached, then it is known as a nucleotide. Sorry, nucleoside. Nucleoside. So nucleoside will have a phosphoric acid group, acid. Then you will have sugar base. Sorry, sugar. Pentose sugar will be there, and also you will have nitrogen base. So all these three combine together to form the nucleoside. And nucleosides, the phosphoric acid group will be present at the 5th carbon of the sugar.
Sir,
Yeah.
the nucleoside only pentose sugar plus nitrogen base. Sir. I think you have written opposite.
Nucleoside. Yeah, you're right. Nucleoside. Nucleoside is this one. Nucleotide is this one. Good. Thank you. So I am representing the pentose sugar. This is the pentose sugar you are having. And here you have a base at the 1st carbon. Okay, hydrogen. Here either deoxyribose or ribose, whatever it may be. OH. Then you have hydrogen. Then you have CH2, which is attached with a phosphoric acid group. What is phosphoric acid?
H3PO4.
H3PO4. This is phosphoric acid. So here you have a CH2OH. And this OH and OH of the phosphoric acid will combine. And they will eliminate one water molecule. This is how they are attached with the 5th carbon. So these three things together, what do you call?
Nucleotide.
It is known as nucleotide. Nucleotide. And if this phosphoric acid group is not present, then what will you call?
Nucleoside.
Nucleoside. Okay. So I hope you got the difference between nucleotide and nucleoside. Did you get?
Yes, sir.
Yeah. Now, these nucleotides, there will be large number of nucleotides, and they will joined by an ester linkage between the uh, between the 5' carbon and 3' carbon atoms of the pentose sugar. Then that will be leading to the formation of a polynucleotide. Polynucleotide. And polynucleotides are nothing but nucleic acids. Nucleic acids are nothing but polynucleotides. Now I will show it again. Here you see these are the different nucleotides present. Nitrogen base, this one phosphoric acid group, nitrogen base, and different nitrogen bases present. Okay, these are the cases in case of DNA. These are the cases in RNA. Now here there was one OH group. This OH group, here you can see first position, it is attached with the base. Fifth position, it is attached with the phosphoric acid group. Now this OH group present at this position, they will combine with the 3' position, this OH group of another molecule, and they will form a diester linkage. Instead of C double bond O, you will have a phosphoric acid group. So we call it as phosphodiester linkage is formed. Phosphodiester linkage is formed, and that phosphodiester linkages will lead to the formation of nucleotides or polynucleotides. So for your convenience, I will represent it again. You will have a sugar base, and in this sugar, or I will represent sugar like this only. In this first position, you have a nitrogen base. This you are having an OH group. Here also you are having OH group or hydrogen is present. Now here this CH2 O P double bond O OH and another OH group is present. Okay, here hydrogen. Now this OH group will combine with the third carbon of another sugar molecule. Here base is there. Here other things remain same. So this is third carbon. This OH group, here OH group is there. They will remove one water molecule between them. And here you have CH2. Then another phosphoric acid group. O, P, double bond O, OH. Then OH. This OH will combine with another OH of the third carbon of another nucleotide. Likewise, it will go on. So that chain will extend. Now see here, phosphoric acid is there, and it is forming P double bond O. So we call it as this group is known as phosphoester linkage. Ester linkage. So how many such ester linkages are formed? One between with this one and one with this one. So we call it as phosphodiester linkage is present between two nucleotides. Phosphodiester linkage is between two nucleotides, and this phosphodiester linkage is between what? Phosphoric acid group of the fifth carbon and OH group of the third carbon of a sugar. And as a result, you will have something like this. Sugar is there. Here you have base, then phosphate group, then you have again sugar. Here you have base, then phosphodiester linkage. Then you have sugar. Sugar is attached with the base, then phosphodiester linkage. So this chain, which consists of large number of nucleotides. So this is what we call as a nucleotide, is not? So it consists large number of nucleotides. So this large number of nucleotides will be known as what? Polynucleotide. Polynucleotide, and polynucleotides are present in nucleic acids. Nucleic acid will contain polynucleotides. I hope you are getting this thing. It is more clear now. Is it so?